class, it's Miss Dunn here, and we're going to pick up where we left off in class today. So you guys just made a video about this, and so this is the Trpoperon. Remember, this is only found in prokaryotes, and let's check it out. So the repressor is sort of continually bound here because the chances of a bacteria growing on lactose in their medium, it's fairly rare. So, but as we know, bacteria does grow on milk and cheese and all these different places. So they do contain these genes in their genome. All right, but we don't really want to use these genes unless lactose is present. So normally we're not going to be able to copy these genes, but if there is lactose, remember this is the sugar found in milk, if this sugar is around, it's going to bind to the repressor and it's going to come off, okay? So what we say here is that this is an inducible operon. Okay, so it's normally off, but we can kind of induce it to turn on. All right, so once this lactose actually binds here, it's going to go away, and we're going to be able to make those, the mRNA, so that we can make these enzymes, okay? So these lac genes contain the recipe of how to make lactase, and lactase enzyme breaks down the lactose. Okay, so the mRNA is going to come out, it's going to land on the ribosome is going to be able to produce these lactase enzymes, okay? So these are the enzymes that are going to go over and break down the lactose. Okay, and then, okay, and then remember that once that lactose is broken down, that includes the one that's on the repressor. So the repressor will go back to the DNA, attach again, and prevent any further production of lactase. Okay, so that's the Trpoperon. I want to talk about one other one. Okay, and this one is called the Trpoperon. Okay, so the Trpoperon, this is called a repressible system, or a repressible operon. Okay, so what that means is that normally these genes are turned on, okay? So turned on normally, but switched off sometimes. Okay, so that's a little bit different from the Trpoperon. The Trpoperon is normally switched off, and then as soon as lactose is present, it turns on. So this is sort of the opposite. And so let's talk about what this is, okay? Everybody has kind of heard of lactose, but not very many people have heard of tryptophan. So TRP, this is an amino acid. Okay, and it's a really important amino acid. You get it from food, but sometimes it's not in food. So sometimes your body has to make it itself, okay? So tryptophan is very important and we need it, okay? Now, we can take certain precursors, certain molecules that are very present all around us, and we have enzymes that are going to produce this tryptophan, okay? And so these enzymes are going to work in a biochemical pathway to produce tryptophan. Now, as you guys might remember from when we talked about enzymes, enzymes can be allosterically inhibited, okay? So this feedback inhibition is allosteric. So if you imagine this enzyme is going to fit around these precursors and change them into the next thing, right? So tryptophan itself can come in and bind here and deactivate this enzyme, okay? So if there's high amounts of tryptophan, that's going to get in the way of this enzyme. So it's going to bind somewhere other than the active site and make it so that this enzyme stops grabbing these precursors, okay? So normally we make tryptophan, but in high amounts we turn it off this way, okay? Now, so one way we can control things and keep homeostatic levels in the cell is by turning on and off enzymes like this. But at the same time, we need to prevent those enzymes from being made if we don't need them, right? All right. So not only do we control the existing enzymes, but we actually go up and make sure those genes become deactivated, okay? And so we regulate the gene expression. So we regulate how much mRNA is being made. All right, so let me show you how this one looks, okay? At first glance, it looks very similar. We got the promoter, operator, and 3 genes, okay? So these genes code for the enzymes that produce tryptophan. Okay, so normally this whole thing is on and we're going to start to pump out tryptophan producing enzymes. So our mRNA transcript is going to get translated and we're going to make all of these enzymes that are needed to make tryptophan. Okay, all right, so these enzymes are going to work together to make tryptophan. Okay, so these are tryptophan. Okay, and these are, remember, this is an amino acid that we need. Okay, we're going to produce that. All right, so let's say we produce so much tryptophan that we're all good. We don't need any more. All right, so at high concentrations of tryptophan, the tryptophan is actually going to go over. It's going to bind to the repressor, and it's going to go from inactive, and it's going to get activated. And now the repressor actually goes on there, okay? So the tryptophan actually kind of activates this repressor and makes it so that it blocks this thing, okay? And so now RNA polymerase cannot bind, okay? And so later, as our body starts to use up all the tryptophan and we need more, your body will actually grab this, or your cell will grab this tryptophan and use it up. And if it does that, then the repressor will come off, we'll, transcribe and translate these genes, and we'll get it going again, okay? So that's the end of that, and go on to the next video.